Hej och välkomna tillbaka till Erik Penser Bank och vår temadag cancerforskning i frontlinjen. Det blir dags för dagens sista bolag att presentera och det är BioInvent. Och för att presentera bolaget har vi bolagets vd Martin Welschoff. Welcome Martin.
Thank you, Klaus.
Take it away, please.
Okay, then I will share my screen.
Exactly.
And you can maybe confirm whether you can see it. I go into presentation mode. Can you see it?
Yes, I do.
Very good. Okay, thank you very much. I'm very happy to participate in your corporate day. And I will give you today an overview where we are with BioInvent. And, you know, there has been a lot of exciting things happening over the last couple of months. So just the forward-looking statement, and then I would like to kick off with a quick snapshot of the company. So we consider ourselves as one of the leading immuno-oncology antibody platforms, and we are specifically focusing on overcoming tumor resistance. Our current lead product, BI-1206, which I will explain to you in a minute a little bit more in detail, is currently in a phase 1/2 clinical trial for relapsed or refractory indolent non-Hodgkin lymphoma. And we just kicked off a second trial, so just around the summer for solid cancers, but I come back to that later in more detail. The company is highly integrated, so we have an antibody discovery as well as target discovery platform, but also, as you know, robust in-house manufacturing facilities. What we are really pushing forward is a portfolio approach. So we have a very robust pipeline and currently 2 programs in the clinic. By the end of the year, 4 programs. By half year next year, probably 5. But I come back to that also later in more detail. The platform, which is highly differentiated, has been validated through Pfizer, with whom we have a very well ongoing collaboration. And this is focusing on tumor-associated myeloid cells from the tumor microenvironment. Just recently, we did a very interesting fundraising, and I have one quick slide on that. So we raised roughly around $70 million before transaction costs, and we did that in 2 steps. We did first directed share issues, and we brought new investors on board, such as HBM, Swedbank Robur, Invus, but also the existing shareholders such as Van Herk, Omega, AP4, and Handelsbanken participated as well. And then that was followed by a rights issue in order to provide everybody who couldn't participate in the directed issue also to recover, basically. The proceeds are mainly intended for pushing our lead program, BI-1206, and then advancing 3 additional compounds into the clinical development. And as I already mentioned, so this will be explained later in more detail. And of course, there will be always some underlying development of our prioritized preclinical programs. This is the cap table of the largest shareholders. So it's Van Herk. They have 13%, Omega roughly 11%, then followed by HBM, AP4, then also Merieux, they have around 4%, then Avanza, Swedbank Robur, Pfizer, so they are more than 50% of the owners, and they represent obviously high-class, long-term institutional investors, which I think is very good for the company. So, as I mentioned already at the beginning, so we're focusing on our portfolio. So we have a portfolio approach. We are not putting all eggs in one basket. So we have a number of clinical programs and such also have multiple shots on goal. We're quite focused on the tumor microenvironment. So we have 3 programs around the target, which is called Fc-gamma-R2b, which I will explain to you a little bit more in detail later. And there we have 2 different antibodies. One is BI-1206 in combination with rituximab, which is currently in a phase 1/2 clinical trial for non-Hodgkin lymphoma. And with the same compound, we have started a second trial in solid tumors in combination with pembrolizumab. Then there's a second antibody called BI-1607, and this is scheduled to go into the clinic at some time point next year. Then we have our in-house screening on T-regulatory cells, and there we have two very interesting programs. One is BT001, and this we will develop in collaboration with Transgene. There we develop our proprietary anti-CTLA-4 antibody in combination with the oncolytic virus platform from Transgene. And then the second one is actually BI-1808, which is a very interesting antibody targeting TNF receptor 2 that we have also identified and discovered with our proprietary screening platform. Both programs are scheduled to start recruiting by the end of the year, which we're really looking forward to. And then we have a couple of other programs, of course, underlying. The most prominent one is BI-1910, but this is still in preclinical development. And then obviously we have the Pfizer collaboration. I will now, in the following couple of slides, explain you a little bit more in detail about some of the programs. And before I do so, just a quick look on our proprietary screening platform, which is patient-centric and the phenotypic screening platform, which means we screen directly on human tumor tissue or human tumor cells, which are directly coming from the patient. We select specific antibodies and then check those antibodies, whether they have therapeutic effects. And if they have so, then we also identify the target. And this is what we use to fuel our own portfolio. And that's also what we're using successfully in the Pfizer collaboration on tumor-associated myeloid cells. So I will start with BI-1206. And BI-1206 we are currently developing in two different indications. First of all, in non-Hodgkin lymphoma, and I will explain that to you in a minute. Secondly, we started a program targeting solid cancers. And the opportunity is really here to enhance established antibodies such as rituximab for non-Hodgkin lymphoma or pembrolizumab for solid cancers. Starting with the target, maybe first, just a quick recap. So obviously you're very familiar with so-called T-cell checkpoints, most prominently CTLA-4 and PD-1, which are inhibitory receptors. And then of course you have activating receptors such as CD28 or OX40. We have the same on cells of the innate immune system, so those are T-cell checkpoints, but you have also checkpoints on the innate immune system cells, and those are the so-called Fc gamma R2 Fc gamma receptors, and you have there one inhibitory receptor, Fc gamma R2b, and a couple of activating receptors, and we are focusing on the Fc gamma R2b, which is the only inhibitory receptor expressed on the innate immune cells. And that is basically important. If you take this break away, then you also will activate the adaptive immune response or the T cells. So this cartoon gives you a quick snapshot how it works in non-Hodgkin lymphoma. There you would know that you can treat non-Hodgkin lymphoma with anti-CD20 antibodies such as rituximab that normally would bind to CD20 and then induce the killing of the tumoral B cells. But what happens is that if this anti-CD20 antibody, in this case rituximab, interact with Fc gamma R2b, which is heavily overexpressed on those tumoral B cells, then it will become inactivated and cannot kill the tumoral B cell anymore. What we're doing with 1206, we inhibit this mechanism. And in addition, we also have single agent activity such that we enhance the killing of the tumoral B cells. This has been published in Cancer Cell, where we could show strong single agent activity in comparison to rituximab, but more importantly, that we could also kill cells that are not responding anymore to rituximab. And this we could show in this model. On the left-hand side, you see the control. Here you see rituximab, and then you have BI-1206 alone, and you see some effects there, but the strong effect you get when you combine those two compounds, and that's also what we're showing currently in the ongoing clinical trial. Just before I go on to the clinical trial, very briefly, so we're currently focusing on 3 subtypes specifically. So it's mantle cell lymphoma, follicular lymphoma, and marginal zone lymphoma. And for mantle cell lymphoma, we have also orphan drug designation. And the clinical trial looks as follows. So it's an open label study, and it's divided into dose escalation and dose expansion. currently at the final part of the dose escalation, and hopefully to move into dose expansion towards the end of the year. And the current study results that we also already have published are as follows. So we're still at suboptimal doses. That's important. So we have started with 30 mg, then moved up to 70 mg, and hopefully move next soon to the next level. And there we have basically 3 responses. One complete response, I think, which was very, very encouraging, but also 2 complete responses actually, and 1 partial response. And this is still at suboptimal doses, which means this is very, very encouraging. And by the end of the year, we hope to have an update that we will present at the end of the year, and where we hopefully can confirm this trend. So far, I think so good. So this slide shows you where we're starting with that compound. So it's indolent non-Hodgkin lymphoma, which then can be expanded into non-Hodgkin lymphoma, but also into solid tumors. And there we have also very strong preclinical proof of concept, but also to autoimmune diseases. So wherever you use anti-CD20 based therapy, there's a potential application for BI-1206. We are focusing on cancer. Regarding autoimmune diseases, we have started some academic collaboration to establish the preclinical proof of concept. So on the solid cancer side, as you probably know, we do this in collaboration with Merck. We have started a trial just around summer, which is also an open label study. And we're currently in dose escalation, and we have started to recruit. And we hope to see early phase 1 results at some time point next year. So then coming to our other programs. So we have basically 2 internal screening programs, one focusing on T-regulatory cells, the other one in collaboration with Pfizer on tumor-associated myeloid cells. And on the T-regulatory cell screening, we have derived 2 antibodies. One is BI-1808, which is targeting TNF receptor 2, which seems to be heavily overexpressed on T-regulatory cells in the tumor microenvironment, which is quite important. And as I already mentioned, so this antibody has strong single-agent activity, but also works very nicely in combination with checkpoint inhibitors. The second antibody that we have derived from the T-regulatory cell screening is our proprietary anti-CTLA-4 antibody. And this one we are currently developing together with Transgene. I mentioned it already on the introductory slide. And we combine it with their oncolytic virus platform, which is shown on this slide. So Transgene has a very interesting oncolytic virus platform, which is vaccinia-based, and into which you can clone full-length antibodies, which we have done with our anti-CTLA-4. And what we then do, you target with the virus the solid tumor cells. And when the virus starts replicating, then also the antibody is produced, such that you get a very high concentration of anti-CTLA-4 only in the solid tumor environment. And both the data of BT-001 as well as of BI-1808 has been published at AACR, and we soon also will give an update regarding further preclinical data in the next coming weeks. Both programs actually are scheduled to start recruiting by the end of the year, and we're very much looking forward to do that. Just to mention, obviously, we have the proprietary manufacturing platform, and this helps us to get some additional income. But most importantly, it helps us to get very quickly from discovery into the clinic. So when I joined the company during the second half in 2018, we had 1 program in the clinic. By the end of the year, we'll have 4 programs in the clinic, which gives you an impression of the speed that we can reach through this high level of integration. So a little bit on the news flow. So during the second half of this year, and this is just high level, and we can probably discuss a little bit more in detail later when we have the Q&A. We expect, as I already mentioned, early results from the phase 1 open label with BI-1206 in combination with rituximab. That should be towards the end of the year. We hope to announce additional milestones from collaborations. and obviously we'll drive 2 new programs into the clinic, which is BT001 and BI1808. Then during next year, we will have then early results from the BI1206 in combination with pembrolizumab, additional milestones from collaborations, and 1 additional program which goes into the clinic, which is BI1607. And I think this is actually quite nice, because then you have a couple of clinical programs running, which are probably will hit a couple of very important value inflection points. So this brings me then to the end, and then we can hopefully start Q&A. So I probably will just leave it at the company snapshot, and Klaus, maybe we start the Q&A now. Thank you very much for your attention.
Thank you so much, Martin, for the presentation. And I will start off with, I mean, you, as you mentioned, you are about to initiate a couple of clinical programs, and you already have 2 programs running. I mean, it's inevitable to speak about COVID-19 spread, and I mean, it seems like this could be increased further during the autumn and winter. How do you address this issue to limit its impact on the recruitment for the clinical trials? Is it possible?
Yes, to a certain degree, obviously. So first of all, I would like to say that so far we didn't have much impact from COVID. So we have been lucky. So we're basically exactly on target. But obviously, this is something that we're constantly monitoring, and there might be some impact further down this year. The only way to mitigate is that you have additional sites, but of course this also has its limit, because you cannot just you know, open 10 more sites, because that also will cost a lot of money. But we have some additional sites, and so far, as I said, we have good tractions regarding recruiting, which also is connected to the fact that we have very, very good and supportive investigators, which are the leading KOL in their respective fields.
Yeah, perfect. And about the 1206 trial that is ongoing in non-Hodgkin's lymphoma, as you mentioned, you intend to. I mean, you're closing in on the phase 1 part of this trial. Do you intend to present this data during a conference this year, or how should we expect this to be communicated to the shareholders?
Yeah, basically, probably we will do 2 things. First of all, we hope to present some data at ASH, which is at the end of the year. But in addition, also we'll give an update, probably in the call with shareholders and analysts, where we give a short presentation. Probably we'll have a press release before, and then have a meeting, have a call, where we have a short presentation and do a Q&A. Okay,
and the next part of this study, do you expect this to be initiated during 2020?
Yeah, if everything goes on well, this could happen. So basically, as I said, so the responses that we have seen so far are at suboptimal doses, 30 mg and 70 mg. Based on what we have seen, receptor occupancy, we think we're close to the optimal dose. So we have already 100% receptor occupancy, but we would like to extend the duration of the receptor occupancy. So we expect to go probably 1 more dose, and then we should be there. But obviously this is data driven, and what I'm telling you today is based on the data as we see it today. But we are close.
Yeah, sure. I understand it. And also about what you listed as upcoming events in this year, it was a collaboration milestones. Could you elaborate a little bit what that could be? Is it from the Pfizer collaboration, or is encoder programs that you expect to advance?
It's probably both. So if you remember from 2019, there we had 2 encoder milestones. One was from Mitsubishi, the other one from Takeda. And further, we're selecting 2 targets, and we expect something similar also this year. That means maybe some encoder milestone, plus also that Pfizer will move ahead with further milestones. Absolutely.
Should we expect that Pfizer are still actively looking for perhaps a third target, and a fourth and a fifth that has been on the table before?
Yeah, so basically what they have done, they have selected 2 targets. So that was last year. And just to talk about those 2 targets briefly, so the next step would be then selecting the respective antibodies, which will be also very interesting milestones from a monetary perspective. Yep. And that could happen hopefully late this year. And then they might also select further targets, so it could be both. Okay, perfect.
And then I would jump to your asset BI-1808. You're about to start a trial in solid tumors, but also in CTCL. Maybe you could just give us some extra color to why is CTCL an interesting indication for this compound?
Yeah, so basically it's wherever TNF receptor 2 is highly expressed. As I already mentioned at the beginning, so it looks, based on the data that we have generated so far, that TNF receptor 2 is highly expressed on T regulatory cells of the tumor microenvironment. And then we have looked there at various indications where you have this correlation very, very high. And that's what we're focusing on. So obviously, you know that T regulatory cells are highly inhibitory, especially in the tumor microenvironment. And if you can deplete those effectively, you would increase very much the response of your own immune system. And that's also what we have seen in preclinical models. So basically what we do is that for all the indications that we pick afterwards, we do also a good, solid preclinical validation. And we presented that data at AACR, and hopefully we'll present later this year an update on the preclinical data. And that basically drives then the selection of the indication for the clinical development.
Okay, perfect. And I mean, 1808 is a blocking antibody of the TNF receptor 2, but you also have 1910, who's an agonist. Could you explain to us the difference? Because it seems like you want to achieve the same goal.
Yes. So the mode of action of those 2 antibodies are quite different, and I cannot explain much more than what we already have disclosed. So I can talk about the effects. Both antibodies have very strong anti-tumoral effects, as well as single agent and in combination with checkpoints, at least in the preclinical models that we have checked so far. And this has something to do with the TNF receptor biology and then the specific antibodies. But both effects are anti-tumoral, even if it should not be clear right from the beginning, because one is an agonist, the other one is an antagonist. But the effect is still anti-tumoral. Yeah.
Okay, great. I just wonder a little bit about, I mean, you have this growing pipeline, but also a strong financial position, of course. When it comes to in-house basic research and trying to find and identify new promising targets, does this work continue as it did before, or are you down prioritizing to add more programs and push them forward?
So basically, we still, even though now we have a strong financial position, so our strategy, at least currently, has not changed, which means that we want to have a portfolio approach and also that we are open to partnering, either geography focused or worldwide. And this is still very much a high priority for us. Coming back specifically to your question, so we will continue also with discovery of new targets and programs, because once we have partnered one or the other, then of course there's room for more. And eventually, at some time point, and this will be very opportunistic, we might also choose a program where we feel there's a good strategy which suits a small company such as BioInvent, that we can also go further. That means show even more than proof of concept, maybe go also through a more broad phase 2 clinical trial. But I think for the time being, where we are focusing on filling our portfolio, showing proof of concept and trying to find partnerships. Saying so, going forward, we might also choose one or the other program that we want to push further by ourselves.
Understandable. And to continue the partnering part, I mean, you've been out there quite a lot with business development since you started at BioInvent. I mean, how is the appetite of innovative oncology assets out there for the moment?
I think it's good, and we have ongoing discussions. I can't disclose much more. I think what has happened, obviously, since March, since COVID is out there, things take a little bit more time because you don't meet companies directly in person anymore, like we do this presentation also digitally. So that means that you take a little bit longer in those partnering discussions, but the appetite is there. And as you said, so I made a lot of good contacts before COVID on which we are currently working and hopefully, bringing one or the other to a conclusion at some time point.
And also, I mean, if you just could give us an update, I mean, what is the partnering strategy for the different assets, or is it? I mean, you don't have perhaps a clear a strategy that is written in stone. But what is? Do you have any assets that you really would prefer to out-license earlier in the development?
At the moment, I would say all programs that we have are equally strong, at least based on the preclinical and early clinical data that we have currently. And we have discussions around all programs, even those programs that are not in the clinic yet. So we'll start recruiting soon. And the strategy is very simple. So obviously, since we're a small company, we have to focus. So when we're just focusing on the 4 major markets, so it's the US, Europe, Japan, and China. And normally what you would do, that's at least what I have done in the past, that you do geography-focused deals first, because, like this, you still keep a lot of potential of the program for a rest of the world deal. But also what you could do in order to, you can achieve a geography-focused partnering, you do, obviously you bring some money in, which is non-dilutive funding to the company, but also you bring validation to the program. And it normally, naturally, of course, the further you're on the clinic, the more interesting is the program for partnering discussions. Saying so, we have discussions around 1206, BT001, BI1808, all of them. Okay.
And when you're talking about geography deals, would that be primarily geographies in Asia?
Yes, most likely, because if you still want to keep enough appetite or interest into a rest of the world deal, once you have given away the US or Europe, which are the largest markets, and even for some pharma companies, Japan is very interesting. So it would be most likely deals focused on the Asian region, if you think about geography-focused deals first. But saying so, it could be, of course, all the other way around. So it really depends on the structure and on the terms that you can achieve at the end. But normally you would start there.
Yeah. And like the deal you did with Pfizer for a couple of years ago, are you interested to do more such deals, or would you rather just focus on your programs?
No, we look at 3 areas. Obviously, the third one we normally don't highlight so much, but we have once in a while manufacturing partnerships, which of course also bring money to the table. So that's the third area. And the first two areas are clearly, so obviously now since we have a portfolio where we have at least good preclinical validation and also some clinical validation, if you think about 1206 in non-Hodgkin lymphoma, that's one of the first two areas. And then, yes, absolutely. So obviously we are currently quite busy with Pfizer and we have extended the Pfizer deal. And what we're trying to do now is obviously to maximize the output. That means the more targets Pfizer selects, the more potential is also there that they select the subsequent milestone, which means antibody selection. And of course, then at some time point, those programs will go into clinical development at Pfizer. So that's something that we're prioritizing, but we're also exploring and have discussions on the platform as well. Absolutely. So to summarize, it's the discovery platform, it's the portfolio, but it's also the manufacturing capabilities. So three areas where we can do commercialization.
Okay, and just my final question then, it's also related to deals, of course. What has been the most challenging part to deliver or to close a deal with a partner?
Well, in what sense? So obviously we have discussions, and I obviously cannot give you the update where we are with those discussions. So
I just wonder if how challenging it is to be out there with really innovative drugs. Is that something that makes it hard to close a deal, or maybe something like that?
Yeah. Okay. So in principle, there's a high need for first-in-class programs, very clearly, because you know, you have the usual products out there and people are keen. And then, of course, it's always the balance between first-in-class and then first proof of concept. So sometimes you can do preclinical deals because a certain company is very, very interested in a very specific biology. In other cases, you need more data in order to have at least some clinical validation, because if it's first in class, of course, then the target as well as the compound is not clinically validated yet, which represents a certain risk. That's also why internally we're not focusing on just one asset, but rather on a portfolio. So it's always the balance between very interesting, very new, but then also they want to see at least some validation. And normally what the companies and I mean the larger partners, what they prefer is clinical validation.
Okay, great. Thank you so much, Martin, for your presentation.
Thank you.